Every detail regarding inherited disease or the mechanics of evolution exists as a set of physical molecules locked tightly inside the nucleus of our cells. To read these sequences, we cannot simply peer through a microscope. We break open the cell walls, extract the raw material, and isolate it for the machine. We strip away the surrounding proteins and cellular machinery, leaving behind nothing but pure, twisting double helices of DNA in a controlled chemical state. Understanding these genetic instructions requires us to physically pull these molecular strands out of their organic housing and prepare them for a digital environment. The sequencing machine has a limited reading window. No current sensor can scan a 3 billion base pair human genome from end to end in a single pass. To bypass this, we utilize intentional fragmentation, shattering the massive DNA strands into thousands of short, readable pieces. This creates a new problem. We are left with a chaotic soup of random segments, and the sequencing machine requires a uniform way to identify and stabilize every piece. We attach specialized Y-shaped molecules to both ends of every fragment. These act as universal molecular handles, or adapters. By cutting the DNA down to size and equipping the pieces with standardized docking stations, scientists convert a messy biological sample into a uniform, machine-ready collection known as a sequencing library. We then wash this library over a flow cell. A flow cell is a microscopic glass landscape covered in a dense forest of synthetic receptor molecules. Watch how this single DNA fragment drifts down and uses its Y-shaped adapter to snap into a secure, upright position on the glass surface. At this stage, we run into a visibility hurdle. Sequencing machines read DNA by photographing tiny flashes of fluorescent light, but a single strand of DNA is so small that its light is too faint for a camera to detect. To solve this, we use bridge amplification. The anchored DNA strand bends to form a bridge with a neighboring receptor. An enzyme copies it, and both stand upright. This repeats exponentially, expanding outward to create a tightly localized cluster of identical strands. Bridge amplification turns a single, invisible molecule into a concentrated beacon of light that the machine's camera can finally record. To read the sequence, we must build a complementary strand of DNA letter by letter. But if we flood the flow cell with normal DNA building blocks, the enzymes copy the strands too quickly for any sensor to capture. The solution is a reversible terminator nucleotide. It carries a glowing fluorescent tag and a solid blocker cap. When it attaches to the growing strand, it emits a specific color. The cap prevents other nucleotides from joining, forcing the reaction to pause. While the reaction is frozen, a laser sweeps across the flow cell. A camera records the specific color of the flash to identify whether the new letter is an A, T, C, or G. Once captured, a chemical wash clears away the fluorescent dye and the blocker cap. The reaction unpauses, allowing the very next letter to attach. By forcing the biological process to stop and wait after every single base pair, scientists gain the precise control to accurately decode long-form genetic sequences. This image shows a digital sequence of A, T, C, and G letters overlaying an organic chemical structure. This represents the moment where millions of analog chemical flashes are logged by a computer and converted into digital text. For quality control, the machine also assigns each digital letter a Phred score. This is a metric that tells us exactly how confident the camera was in that specific color flash. The genetic letters and their corresponding confidence scores are then bundled together into a standardized data file known as a FASTQ package. The creation of the FASTQ file marks the transition point of this process. A physical, biological specimen has been successfully converted into digital information. However, a single FASTQ file is only raw data. It contains millions of short, jumbled fragments that now require computational power and bioinformatic algorithms to align and assemble. This flowchart demonstrates the downstream pipeline. Raw reads are mathematically aligned to reference genomes, plotted onto gene expression heat maps, and structured into scatter plots for analysis. These computational pipelines allow us to map human genomes, track the mutations driving cancer, and analyze the complex microbiomes that dictate our health. We have built a continuous process that runs from physical extraction in a wet lab all the way to algorithmic processing in the cloud. The physical sequencing journey is complete. Now, the computational journey begins. Welcome to RNA sequence analysis, from sequencing to preprocessing.